Thanks. So my name is Alex Bloom. I'm the Chief Technology Officer at AviadoBio, so I'll spend the next 10 to 15 minutes giving you a bit of background about who we are and what we're doing in the space of CNS gene therapy. So we are a UK headquartered gene therapy company, and it's our mission to develop AAV gene therapies for CNS disorders, starting in the neurodegeneration space. We were founded out of King's College London with input from the UK Dementia Research Institute. We're around 60 FTEs at the moment, the majority of those in the UK, but also with some of our team in the US as well. Our focus is around AAV gene therapy delivered in vivo, and what we're looking to do is to maximize biodistribution, so to really think about how best to treat different CNS disorders by targeting the right structures in the brain and in the spinal cord to generate the maximum clinical effect with a tolerable safety profile. And we can enable this by the multi-functional expertise in the company, vector design, CMC, and our clinical development and translational teams. Our lead program is termed AviadoBio. This is a AAV9 gene therapy, which is currently in clinical trials for a aggressive dementia called frontotemporal dementia. We're currently recruiting patients in that study at the moment, presented initial safety data last year at ISFTD. And then we have a pipeline of earlier stage assets targeting much more common neurodegenerative disorders using both gene supplementation and using gene silencing. To date, we've raised a significant amount of money in our Series A and partway through our Series B financing, and we have a partnership with Astellas Pharma for AviadoBio. This is the leadership team driving the company forward, headed by Lisa Deschamps, our CEO. Lisa was at Novartis and led the acquisition of Avexis and the commercial rollout of Zolgensma globally, probably the most successful commercialization of a gene therapy to date. But Graham, David, John, and myself all have multiple years in vivo gene therapy experience. Our founders in the bottom left, all at King's College London, and then we are really privileged to be supported by a broad syndicate of investors, both on the VC side in the UK, Europe, and the US, private equity, and also on the pharma side as well. So this is our pipeline in a snapshot, headed by AviadoBio. I'll talk more about that program in a minute. And then our pipeline behind this, so we're really going after the common drivers of neurodegeneration with our pipeline, MAPT targeting tauopathy, and the SNCA program targeting alpha-synucleinopathy. These go after the most common drivers for Alzheimer's disease and Parkinson's disease, and we use vectorized gene silencing. And then behind that, TDP-43 proteinopathy, gene replacement strategy there. This targets 97% of ALS. So moving on to AviadoBio, which is our lead program in the clinic. As I mentioned, this is an AAV9 gene supplementation therapy. So we look to supplement the progranulin gene, the granulin gene to make progranulin protein in a subset of frontotemporal dementia patients. So frontotemporal dementia is a aggressive and early onset dementia. So patients typically present in their 50s or 60s, but we've seen patients present much earlier than that, sometimes as young as in their mid-30s. And it's an aggressive dementia, so from disease onset to death, the average is about 7 years. The disease biology is very well established in FTD-GRN, so this is a haploinsufficiency disorder, so patients have one functional copy and one non-functional copy of the granulin gene. This leads to a haploinsufficiency of the progranulin protein, anywhere from 30 to 50% of normal levels. And people tolerate that through life, and then there's a trigger point later in life where they can't tolerate anymore. Their lysosomes become dysfunctional, and this drives neuronal sickness and ultimately neuronal death and neurodegeneration. So a simple strategy here, we're looking to restore progranulin back into its normal range in the CNS, particularly in the tissues that are most affected by disease, so the cortex and the cortical regions, to hopefully slow disease progression, potentially even halt it. There's a very tractable clinical development plan in FTD, so it's a very well understood and characterized disease lots of established natural history cohorts, and really good endpoints, both from a biomarker perspective, looking at things like volumetric MRI change and inflammation markers, but also the harder clinical endpoints around cognition, so things like GenFCog and CDR-NAC scoring. So how do we do this? So we've taken a different approach to others. We are looking to deliver our AAV9 gene therapy directly to the brain using intraparenchymal delivery. And we deliver the therapy to a region of the brain called the thalamus. And we do that very deliberately because the thalamus is a key hub, it's a central relay hub in the brain and is very well connected. So we can use the thalamus as a factory to make progranulin protein that then gets transported throughout the neuronal networks, and you can see these in the image here, throughout the brain. And we've demonstrated this process of production in the thalamus and transport to distant regions of the brain in multiple animal models. So I don't have time to go through all of the data today, but we have run, I think it's around 10 animal studies to support the pharmacology and toxicology of AVB101, starting in mice using granulin knockout models. We've been able to demonstrate pharmacology after direct administration, but then much more importantly, using large animals, so our GLP species in non-human primates, but then also the sheep model, which have a brain which is larger than a primate, to model the delivery to the thalamus, the production of progranulin protein, and then the transport of that protein to the cortical regions. And we've been able to demonstrate restoration of progranulin protein to the normal range in the cortical structures, and as far as I'm aware, we're the only company that's been able to show that. So as I've mentioned earlier, we're in an active clinical trial for AVB101. It's called the ASPIRE study. This is a classical phase 1/2 clinical trial design. It's an open label study of a 3+3 design, looking to enroll 6 to 9, potentially up to 12 patients in a dose escalation manner. The primary endpoint for this study is obviously safety. We're looking at treatment emergent adverse events, but also signs for efficacy and target engagement, so CSF progranulin levels, biomarkers of neurodegeneration, inflammation and lysosomal function, and then some of the harder endpoints, volumetric MRI and cognition scores. In terms of the study itself, we are actively recruiting in 8 countries, in North America, in the US and Canada, also in the EU and in the UK. We have fast track designation for this program in the US, as well as orphan designation in Europe and in the US. We also have a sponsored genetic testing program that's operational in Europe, in the US and in Canada to identify patients for the study. And we use what's called a hub and spoke model for the trial. So we have expert surgical centers that can perform the neurosurgery to deliver this therapy, and then patients are followed up locally at their neurological site. And this is how we actually deliver AVB101. So we use intraparenchymal delivery, and we use CE-marked off-the-shelf technologies that have been around for many years for direct delivery of therapies to the brain. We work with a company called ClearPoint, based in California in the US. And actually, this is a path that's very well trodden, and there's an approved gene therapy in Europe called Upstaza, marketed by PTC Therapeutics, that uses the same technology to deliver a therapy directly to the brain in a pediatric neurological disorder. So what we do is we mount an MRI-compatible stereotactic frame that you can see on the left directly onto a patient's skull, and then we pass a very, very fine cannula into the brain to the target region that we're looking for. And it's about the diameter of an angel hair pasta, and we can monitor this under real-time MRI. So we tag our therapy with something called gadolinium, which is a tracer that we can see in the MRI, and we can actually watch as we infuse the therapy into the thalamus. And you can see here on the right, these are some images from one of the patients in cohort 1 of our study, and you can see the sort of white balls, and this is the gadolinium lighting up in the 2 thalamic hemispheres in each hemisphere of the human brain. So as I've said, we have dosed the first 3 patients in the low-dose cohort. We've successfully held a DSMB meeting. We've escalated to our mid-dose for the trial. Really good news that we presented at ISFTD last year was we've seen no SAEs to date, and importantly, we've not needed to use prophylactic or reactive immunosuppression. That's really, really important in neurological conditions where you're looking to measure cognition. Investigators really would prefer against the use of things like steroids that can affect the ability to perform cognitive and functional tests. And we're expecting to report initial functional and biomarker data later this year or early next year. I did mention briefly we have a partnership on this program with Astellas. So towards the end of last year, Astellas took an option for AviadoBio with an upfront payment and just over $2 billion in sort of backloaded milestones. We're really delighted to have Astellas as a partner, as a pharmaceutical company with a deep commitment and actually a deep understanding of the requirements of AAV gene therapy, and they've been very supportive. So just turning now to our pipeline. So I'll talk mostly about our vMix platform. So we're using vMix, which is our own proprietary gene silencing platform to target tauopathies and alpha-synucleinopathies. So this is essentially vectorized RNA interference. And we have spent many, many years, actually, back from the days in Chris's lab in King's through to today, to think about the very best design for this platform. It's very, very important for gene silencing to ensure efficient, high-fidelity silencing with minimal to no off-target effects. And we're now able to demonstrate this going from in silico design, where we apply AI and machine learning algorithms to identify the best regions to target to silence pretty much any gene that we want to, through to in vitro data. We can do that in a matter of weeks. And then what we've seen is that translatability from in silico to in vitro to in vivo. It's highly predictive, and we've demonstrated that now across a range of targets in mouse models, and we're planning to move into large animal and NHP studies imminently. By using things like cell-specific or tissue-specific promoters, we're able to very finely tune expression of vMix guides on a per-cell basis. So we can very effectively target different structures or different substructures or actually individual cell types in the brain to go after diseases in a very, very effective and precise manner. And just to close, we are currently in the middle of our Series B fundraising. So we're looking to target the second close, which is the final close for the round, looking for just shy of 20 million that we're planning to close imminently, certainly in the next quarter. And the use of proceeds from this raise will be to deliver the ASPIRE trial and actually take us to the point where we're ready to initiate registrational studies, to take our tau program into clinical development, and to generate initial biomarker data with our tau program in Alzheimer's disease, and to progress our discovery pipeline. And we have an ambition to have 3 programs in clinical trials by 2027. So with that, I want to say thank you for your attention.